السلام عليكم. يعطيكم العافية. راح نبلش اليوم محاضرات two lectures about bacterial growth. المحاضرات هذولا I have depended mostly on Jawets for the content here. أول شيء بدنا نحكي عن what is bacterial growth. هلا in a given biosphere or a culture or a specific niche or an environment, in microorganisms or bacteria, the numbers of the cells, or the amount of the cells, or the number of the cells, the population of these cells usually remains constant. فكروا فيهم زي ما كيف الجلد بضلوا يتغير ويتبدل. So there are cells that are dying, there are cells that are alive. All of this because why? الجلد مثلا بيجي نقطة معينة أو جزء معين أو عفوا كمية معينة من الدم ومن nutrients. So at this given biosphere عندك هدول الخلايا, they do not outgrow, they try to not outgrow their environment, so they remain constant with this environment. هذا طبعا as long as you are controlling the inputs اللي هو growth factors and the outputs اللي هم toxic byproducts. هلا كل هذا this is due بحكي لك to the fact of the rate of growth of these organisms is balanced with the rate of death. So they are growing, they are dividing in the same rate as they are dying. هلا within a living host, هلا بالنسبة لنا إذا بنحكي عن biosphere إحنا إذا الهوست اللي هو الإنسان, جسم الإنسان is a biosphere. In a particular niche within the host, مثلا في ال GI tract, على skin, بالblood, whatever it is causing its infection, or living symbiotically with you. The survival of a microorganism is dependent on its ability to be successful in competing with the host and other microorganisms for nutrients. شو يعني؟ يعني this bacteria has to be able to acquire its growth factor from the host, has the growth requirements are adequate for its growth, and is able to evade the host's destructive ability. مثلا, it has other bacteria that wants to take these nutrients, other bacteria that may have toxic byproducts that want to kill it, or the body itself that might have immune system that wants to kill it. If it can bypass these things, bypass these hurdles, and then at the same time it can acquire the growth it needs, the growth requirements it needs, معناته هذا صارت البكتيريا هاي إذا تحققت بهذه الشروط, then it becomes a living organism in that area, or مثلا إذا كان هاي الarea does not have other organisms, it will cause an infection. So this is what we're talking about. So consequently, knowing the intricacies, عشان هيك إحنا لازم نعرف details, intricacies, كل هدول البكتيريا, how they interact with each other, how do they compete for food and resources, لكل micro environment بتهمنا. هلا بالنسبة لنا كتير مهم إن نعرف their growth, their survival, and how they evolve during these conditions. هلا our current understanding is usually based from إيش؟ إنه studying bacterial physiology, their growth, while controlling all conditions in an optimal manner. هذا اسمه culture conditions, culturing. So culture conditions in a manner, إحنا مثلا بأي دراسة علمية أنت you control all the inputs and you leave one input uncontrolled and you study that input that is uncontrolled. بالنسبة لنا, what is the input that is uncontrolled? إحنا بدناش البكتيريا. We want to grow, to study their growth. So what do we control? We control everything else. So we control all the conditions that affect its growth. We're going to control we're going to control temperature, we're going to control pH levels, we're going to control salt levels, we're going to control CO2 levels, oxygen levels. We're going to give them excess nutrients. إذا مثلا الحركة القليلة causes them to change their genetic material, we keep them moving. So we control. This is called a culture. هلا in the real world, بالعالم الحقيقي, in جسم الإنسان or whatever, microorganisms do not have all these excess optimal conditions. هذا طبعا it's not true. And realistically, they are living under nutritional stress and every other form of stress. So therefore their actual physiology is influenced by these different stresses. نقدر احنا كمان we can simulate these stresses. إذا بنقلل الأكسجين بنطلع على الجينات بتتغير، شو بتغير فيها، how the protein structure changes, how the transcription changes, and all that. هلا if there is a vacancy in a niche، هاي نقطة مهمة. If there is a vacancy in a niche in the environment, most importantly مثلا a microbe was unable or failed to grow, it will be filled by another usually more competent organism. معناته ايش؟ إذا أنت عندك نيش وأنت استخدمت أنتي بايوتيك وقتلت البكتيريا بهذا النيش، is it going to become sterile؟ طبعا لا. إذا عدا عن حكيت لكم the 3 usually sterile fluids in your body, in any other place, in the stomach, in the lung or whatever, كان في بكتيريا قتلتها, another bacteria will come in its place. It will not be sterile, it will never be sterile. Whatever was there will maybe the other bacteria that was not killed by the antibiotic will fill that niche, or another more competent organism will come. هذا بيتطرق إلنا ل antimicrobial resistance and virulence virulent organisms. So more virulent organism might come, or an antimicrobial resistant organism might come and fill this niche. So we have to be very careful in creating vacancies in certain environments in the body. Sometimes you want just to control, not overly kill. هلا that's why I was هذا بحكي لك اللي حكيناه هلا. Why is our current approach is flawed? You know, if our current approach, you know, we want to destroy bacteria, we have to change this approach. We cannot destroy bacteria. We do not want to be sterile. دائما ماسكين احنا هالانتيسبتكس شو اسمه اللايسول وابصر شو بننظف كل شيء حوالينا. You have to live with bacteria rather than think that you're going to destroy bacteria. You cannot destroy bacteria. The current approach إنه you have to live with it. It's part of your organism. It's part of your environment. You have a competent immune system that will deal with it, that will live with it. So this is part of our evolution. احنا لما صرنا نعرف عن البكتيريا وصرنا نعرف عنها اكثر واكثر واكثر، صرنا نحاول من نقتلها، بدنا we destroy it, we want to remove it out of the but you can see انه مثلا اذا حكينا تذكروا بالمحاضره الماضيه انه مثلا اذا كان عندي مليار او بديش مليار 100 مليار خليه ببكتيريا، كلهم حكيت لها we mentioned how their genome is always ever changing. رحنا احنا قتلناهم، احنا حطينا عليهم كل انواع الستريس and one survived. That one bacteria that survived is able to survive. انت عملت عليهم so much stress that you have selected، انت نقيت البكتيريا that will be able to overcome all the stress. بعدين هاي الوحدة in a given week or 2 weeks or a month it will become 10 billion again or 100 million again. So you have created a superbug by creating all this. So this is the approach that we were talking about. So we are only preparing the place for another group to take over, is what we're trying to talk about. So once in an appropriate niche, bacteria reproduce by binary fission. هلا واحد يسأل شو الapproach؟ الapproach is to try to control rather than outrightly destroy. So an approach, bacteria reproduce in binary fission, by which one parent cell divides to 2 progeny cells and one reproduce 2, and this is exponential growth 1 2 4 8 16 وهكذا. The meaning of growth is the increase in the total sum. شو ايش احنا بالنسبه لنا البكتيريا how do we mention what is growth بالنسبه لنا؟ if all the components of a given organism is increased, the sum of all of them is increased, this is why an increase in size in a given cell is not true growth. مثلا خلية كبرت حجمها وزادت بروتينات فيها, it is not true growth. We want all the components of the organism to increase in size. That will that for us that is deemed growth. Unicellular organisms growth will result in cell multiplication. Growth leads to an increase in number of a single cell bacteria making up a population of this bacterium. This is bacterial culture. So in a bacterial culture, growth is increase in number of these cells. How do we measure these microbial concentrations? How to measure the concentration of a living organism? كيف أنا بدي أشوف تركيز البكتيريا؟ مثلا احنا نشوف تركيز السولت أو تركيز مثلا الدواء، تركيز chemical. Can we see the concentration of our organism? So there are ways that we can do this. we measure the concentration as the number of viable cells per unit of volume. هذا واحدة منهم، هلا بنحكي كيف نعملها. or by mass، نمسك الخلايا كلهم بنوزنهم الماس تبعهم per unit of volume. مثلا في عندي مثلا 1 gram per 1 liter مثلا of bacteria. once I dry them and يزيح عنهم ما يزيح عن كل شيء، نحطهم لحالهم in a pellet and we measure that pellet, basically the weight of that pellet. However, these two types of measures are not equal. So average dry weight can vary at different stages of growth within a culture. راح نحكيها هذا كمان شوي قدام. And each of these measurements is thus used for different studies and application. كل واحدة مستخدمها لapplications مختلفة مش لنفس الشيء. So إذا بدك تدرس genetics and inactivation of microbes، يعني قتل الميكروبات أو cells concentration is used اللي هو الأولى. Whereas قاعد تدرس biochemistry and the enzymes and the proteins of the bacteria, you might want to use the biomass concentration instead. Cell concentration, the viable cell count نحكي عنه هلا بالتابل الجاية is the typical measure of a cell concentration. So an easy way to indirectly measure a viable cell count for most purposes is used by measuring the turbidity of a culture. شو يعني turbidity of a culture؟ طلع هون. هلا الكالتشر عادة مبين هيك، ببلش معاك هيك. So after a while when bacteria start to grow it becomes turbid. So if you can measure the turbidity، طبعا مش راح يبلش يكون هيك بعدين مرة واحدة يصير turbid. It's a gradual thing and then it increases, it plateaus. So if you measure this turbidity, you can indirectly measure the growth of the bacteria. The relationship between the viable cell count and the turbidity is made into a standard curve. This is the cell concentration. With experience, it is possibly أحيانا it's possible to roughly estimate by just looking at it, but that takes about a few years of work with bacteria. A barely turbid suspension مثلا فياته 10 to the power 7. More turbid suspension may contain 10 times more than, and so on. هلا the main drawback of connecting turbidity with viable cell count is that during growth the viability هي مهمة. هلا هالخلايا في turbidity معناته في خلايا، هل كل هاي الخلايا عايشة؟ كلهم هذه احنا حكينا انه الخلايا بتموت وبتعيش. But the viability of these cells is different. مثلا turbidity بعد 8 ساعات إذا كان على ال maximum growth of the cell is not the same is the same turbidity بعد 16 ساعة أو بعد 20 ساعة أو بعد 24 ساعة، لأن الخلايا وصلت لعددها النهائي. بس ال viability بعد 20 ساعة is not the same viability as بعد 8 ساعات. 8 ساعات they are more viable. بعد 20 ساعة they exhausted most of the liquid of the nutrients and there are more cells, but a lot of these cells are dead. So as the cells in the culture reach different balance between rate of growth and rate of death as a result of this balance. Cells may be less viable زي ما حكينا but the turbidity remains the same. هذا اللي حكينا هلا. So إذا كان حطيتهم بدك how do you measure viability؟ إذا بتمسك مثلا هالبروث هذا اللي هنا in A and then you look at it without diluting, you put it on a plate. So you put it on an agar plate and you take a little bit and then you مثلا 1 microliter 10 microliter 100 microliter whatever, and then you try to see how many colonies form out of these. So this is called the plate count. The plate count is when you put a specific volume of a specific culture مثلا هو الculture اللي شفناها هلا, and then you spread it over this agar and you see how many colonies form out of this. Undiluted you get so many colonies you cannot count them. يكونوا زي كأنهم ملزقين في بعض كلهم كتار. بعد dilution by 10 times يعني بس مثلا يعني مثلا أخذت 100 microliter وحطيت عليهم 900 microliter مية. So you diluted it one time. It's still too many. You do that again and then you start getting a countable number and so on. This is called the viable count, and this is how you can determine how viable is your turbid culture. The biomass density, the idea من هنا is to determine the dry weight of a culture. So the bacteria, after it has been washed with distilled water. Why am I washing with distilled water? I am removing all the salts, excess salt, excess nutrients. I want to measure just the mass of the cells by themselves. كيف عادة بنعملهم؟ بنجيب إحنا we centrifuge. When you centrifuge these cells زي ما نحط blood مثلا, you centrifuge and you separate the plasma from the cells in the blood. The same thing when you centrifuge كل الكلتشر بيطلع كل everything, and the cells all of them will pellet بلزقوا مع بعض بصيروا زي كأنهم طابة مع بعض. You wash this طابة, this pellet, you wash it a few times, you remove all the excess salts, excess nutrients, excess everything, and you just make sure you only have cells, and then you wait for it to dry, and then you weigh it, and this is basically how you can see the biomass density of this bacteria. This procedure is even more time and effort demanding than determining the cell viability. طبعا راح نشوف إذا كلها حكي الخطوات بنحكيها. This is actually time consuming. So again, cell mass is correlated with the dry weight with turbidity using a standard curve زي ما عملنا viability الأولى. You can still use تحكي إنه مثلا التربيديتي الفلانية تقدر تقيسها مثلا التربيديتي الفلانية للبكتيريا الفلانية ساوت الماس الفلاني والتربيديتي الفلانية ساوت الماس الفلاني. So without having to do all the steps to measure the biomass, you can just reach a certain turbidity and you can know that I have a mass of so and so. So the standard curve is done for either viability or weight by calibrating each turbidity measure وهكذا ما حكينا هلا. Alternatively, the concentration of biomass can be estimated indirectly by measuring an important cellular component such as protein. مثلا ممكن تقيس البروتين بالهذا مثلا وياها عندنا طرق لقياس البروتين. مثلا تقيس البروتين لحاله بتعرف انه مثلا والله بس عندنا الماس كذا. Well, this is another indirect method. So exponential growth, the growth rate constant. بدناش يعني هذا نزيد عليك. الشيء الوحيد اللي بدنا نعرفه انه there is a specific growth rate constant for each bacteria. So كل بكتيريا هذا it's a first order reaction معناته it's a linear growth. First order reaction بتعمل هيك مثل ماس and ضل بيطلع right away like this. So this is زي هيك هي. So this is a first order reaction. So it's a linear growth that depends on nutrients present. إذا كل شيء present, it will keep growing in that fashion اللي هي high. The rate of growth is calculated. ما بدنا نعرف calculation. A growth rate constant of مثلا 4.3 hours. One of the highest recorded means that each gram of cells produces 4. يعني هذا كثير سريع. So each gram of cells produces 4.3 grams of cells per hour. So this is very very quick. فإذا كان مثلا عندي كان الجروث ريت 0.02 معناته so ال 1 جرام بعد ساعة بصير 1.02 جرام. So this is very slow growing. Why does this matter for us? It's very important لأن بدك تعرف مثلا إنه إحنا إذا بدي يصير عندي infection من بكتيريا في شغلتين بدي مهمين عندي إنه what is the infectious dose, how many number of bacteria do I need to cause an infection. هاي الأولى. بعرف إنه مثلا إذا 500 خلية بتعمل disease معناته I know this is the infection dose. الثانية is the doubling time which is the growth rate. إذا ال 500 هدول قديش بدهم وقت يصيروا 1000. إذا كان doubling time تبعهم سريع معناته the infection will be much quicker and then you will have the symptoms much quicker and you need to react much quicker. بالعكس اللي إذا كان بطيء. وهكذا راح تشوفوا إن الأشياء كتير مثلا في عندك خلايا مثلا tuberculosis infectious dose كتير واطية بس الدبلينج تايم كتير واطي برضه. So it's very slow grower but because of slow growth it's very hard to destroy. We will get to that in time. So the generation time, the doubling time هذا اللي هو بهمنا اللي هو يعني related to the growth rate is required for the population to double. So this is لأنه by binary fission. What is the time for doubling time؟ هذا مهم. مثلا doubling time لل E. coli is about 20 minutes مثلا. So in 20 minutes إذا بتحط 100 خلية they become 200. بعد كمان 20 دقيقة ال 200 بصيروا 400. كمان 20 دقيقة بصيروا 400 يصيروا 800. So in 1 hour you have from 100 to 800. So that's the doubling time that we look at. Usually it's determined by plotting the amount of growth as you can see here. هلا a rapid doubling time corresponds to a high growth rate constant زي ما حكينا. So doubling time of 10 minutes corresponds to a growth rate constant of 4.1 which is very high. 35 hours is 0.02 which is very very slow. The growth curve is the data plotted on a curve. So كل هذا اللي حكيناه we make it in the data. We take data the growth viability, the turbidity, and we plot this on a curve and we get out a growth curve. And you can see that all bacteria have a growth curve. All of them have the same growth curve, but يعني عندهم the same phases but in different rates and different so in different rates basically. So a known fixed volume of liquid medium is inoculated with a microbial cells. مثلا ايش يعني؟ انا بعرف مثلا عندي 5 mil or 25 mil or 50 mil. I have a known fixed volume that I do for all in my experiment for all these bacteria, and then I inoculate them with the same amount of bacteria, مثلا one colony مثلا, and taken from a previous culture that grew to a saturation. عشان أنا إنه هلا الإشي اللي حطيته أنا is one colony. I don't know how much of that colony I added, so I wait for it to saturate, and then I take from that saturated growth curve a known volume into another known volume, and then I start doing my data collection. While at period points the viability of cells is also determined. زي ما عملنا we can do viability count زي ما حكينا بالتابل. So the phases of bacterial growth in a growth curve shown are due to population growth events, not a singular cellular event. تمام. This type of culture is refers to a batch culture. هذا حكينا هلا what we're doing here is a batch culture. Has 4 phases هلا بنشوفها. And you must remember that a batch culture resources are controlled and are finite. مش so we are مثلا بنحط عادة الأكسجين والtemperature is the same, but the nutrients, the resources هنا هم are the nutrients are finite. مثلا حطيت بيبتون وشوجر و whatever is in there is in this batch culture is it. I'm not going to refresh it. So that's how I can measure the growth. قديش بيعيشوا لقديش بوصلوا لما يستهلكوا كل إشي ومتى ببلشوا يموتوا، قديش سرعة الموت، قديش سرعة الحياة، وهذا this is basically the growth curve. هاي هي growth curve. كل البكتيريا they follow the same phases of microbial growth. At first you have a lag phase, so everything is there but they're not growing yet. It's not growing, they're not growing. What is happening here? They are adapting to the new environment. طلعتهم من environment they were saturated، كانوا مستهلكين، كانوا تعبانين or in a different environment, and then this is the time for them to recondition their genome to the new environment. So they are first lagging. بعد after they are lagging they start growing. هلا بصيروا يستهلكوا كل this is doubling. So مش حكينا doubling is linear first order, this is the first order doubling. So log exponential growth ضلوا عايشين لما يوصلوا مرحلة they hit a stationary phase. Why? Because هنا توصل هاي المرحلة بطريقتين، يا انك استهلكت النوترينتس or صار في عندك تجمع لباي برودكت that is too toxic for growth. فهذا بهمنا لما انا ارجع اقتل البكتيريا in مثلا with a drug. بهمني انا مثلا انه I know what is a byproduct that I can give that I can send them to a stationary phase, or what can I do to prevent them of growing exponentially. How can I target them here? How can I target them here? These are basically لما نحكي عن antimicrobials, this is basically what we're talking about. This is the very important piece of information that we always depend on. لما يوصل stationary phase, accumulation of byproduct, or they have exhausted their nutrients, بصير the rate of their growth equals the rate of their death. So they are back to a stationary phase, back to no growth, but there's no excess. التربيديتي ما بتزيد والviability تصير constant. بعد فترة they start going into rate of decline. هلا إذا شايفين rate of decline is slower than the rate of growth. So they are holding on to dear life as much as they can. Here they are growing as fast as they can. Here they are trying to die as slow as they can. So they start recycling material, recycling cells, doing as much as they can to maintain their growth, to maintain their as to keep ولو خلية واحدة ضلها عايشة. Because if that happens and nutrients are added here, what's going to happen? They're going to go back to an expansional phase again. So that's what they're trying to do. They are communal. They try to live together. They try to maintain growth. فهذا اللي حكيناه كله. I think the lag phase is the phase where bacteria from the previous culture are now exhausted. حكينا كانوا تعبانين، ما عندهم metabolites، ما عندهم الانزيمز اللي بحاجتهم. So they need new enzymes. They need new start new make new proteins. So كل هذا بده وقت. This is why they are lagging first. So that's their difficult starting out. They want to catch their breath. Remember what مثلا حكينا spore forming condition كانوا تعبانين كتير and they get that phase they start making spores مثلا. So once they are in the favorable condition بدهم يعمل new intermediates as we mentioned, and an even longer lag phase happens if we switch from a different medium. إذا تغيرنا الميديم كله كانوا عايشين على جلوكوز حطيتهم منطقة مثلا ما فيها جلوكوز فيها لاكتوز whatever something else that they still can use. بدهم still معناته هون بدل انزيم واحد بدهم يعملوا انزيمين اثنين أو ثلاثة whatever. So the more changes you do to the media, the different the lag phase is going to be longer. So it's not only protein conditioning, again that means it's at the gene level conditioning. The exponential phase, during the exponential phase the cells are in a steady growth state عايشين بسرعة بيعيشوا بسرعة. New cell material is being synthesized at a constant rate and the new material is itself catalytic. The steady increase will continue forever. إذا أنت بضلك تعطيهم nutrients forever, they will keep in this exponential phase forever. Until one of the two conditions حكيناها الأولى إن nutrients has been exhausted or the toxic metabolite has been accumulated to inhibit growth. Aerobic organisms راح تشوفوا إنه the nutrient that becomes limiting لهم هو الأكسجين عادة they exhaust the oxygen right away. فهذا إشي مهم بالتيث مثلا لما نشوف مثلا إنه بنحكي عن in teeth and how the anaerobes start to grow there, how the facultative anaerobe they can use oxygen and non-oxygen. So once they deplete the oxygen, they can switch off, and then they can improve the condition for anaerobes, which would become anaerobe favorable for anaerobes. This is one condition مثلا. So in case of bacteria, the growth rate will decrease unless we force oxygen. هذا مثلا, that's why we keep rotating the culture to make sure that the oxygen remains constant. عشان بس تضل nutrients are. So when the bacterial concentration reaches مثلا, we can no longer introduce oxygen sufficiently because of the cell concentration is عالي كتير إنه ما بتقدر تدخل الأكسجين. هلا وصلنا stationary phase. Eventually the two conditions put you in a stationary phase, and in this phase the cell turnover is the status quo. اللي هو this is where there is slow loss of cells, but then there's still slow growth of cells, and some material is being recycled. When this occurs, the total cell count slowly increases, and although the viable count stays constant. بعدين بدخلوا بالdeath phase. After a period of time ما بيقدروا يضلوا بهالconstant phase على طول. They start to lose cells. So the death rate increases until it reaches a steady level. The rate of cell death is much slower than that of exponential growth. Cells are already adapted. They can recycle, turn into resistant, less active form. هاي نقطة كتير مهمة. This is the death phase. This is the phase where they want to make spores if they are spore forming. If they are أغلب البكتيريا مش حكينا نوعين هم اللي مثلا gram positives اللي هم bacillus والclostridia that make spores. But other bacteria they don't have spores, but they can form something like spores اللي هو they're resistant active, less active form. So they hibernate basically. So they try to hibernate. That's how they reduce their rate of death. So after the death phase, most of the cells have died and the rate of the death dramatically decrease. بحكي لك مثلا this helps a few number of cells persist for almost years. فهذا هو this is the secret of bacteria, how they can maintain themselves on surfaces for a long time. This persistence is due to cells turning on genes that helps them survive زي ما حكينا عن spores مثلا. فهون النقطة هي viable but culturable cells. إنه مثلا إذا بدخلوا بهاي الفيز you try to culture them but they do not get cultured, but they're still viable. It's because they are dormant and sleeping basically. So this is basically a genetic response to extreme stress. Maintaining cells in the exponential phase. For most viable cells, a continuous culture can be made. إذا بدك تدرس الخلايا in exponential phase, so you have to remove the two conditions that send cells into the stationary phase اللي هم toxic byproducts and the exhaustion of material of nutrients. فشو بتساوي؟ بدك تضلك تضيف nutrients وتضلك تشيل ال toxic material. That's how you maintain cells in exponential phase. في جهاز هذا اللي هو اسمه chemostat. Chemostat is basically في له siphon outtake ضل يسحب من هالكلتشر تبعك وياخذ منه التوكسيك باي برودكتس، من فوق كل ما بسحب نقطة بضيف بدالها نقطة فريش ميديا من تحت. ف this way you maintain the culture in a concentration that does not have enough enough toxic byproducts to send cells into the stationary phase, or enough material still is fresh material موجودة عندهم nutrients that they're still in exponential growth. So the vessel is inoculated and the cells grow until the limiting nutrient مثلا is exhausted at an equal rate of consumption. تمام. and this is the chemostat. هاي من فوق بضلني أضيف new fresh material، ومن تحت an outflow of media and microbial cells to the collection vessel. بضل أسحب منه هون التوكسيك باي برودكتس وأضيف اللي هو الماتيريال. دائما بضل أحط عليهم الاير وأحركهم، أعطيهم التمبريتشر اللي بدهم إياهم والبريشر اللي بدهم إياه والبي اتش اللي بدهم إياه، كل شيء بعطيهم إياه. In this manner I maintain them in exponential phase. And it's important to study them in an exponential phase because this is the phase where they are يعني مثلا خلايا لما يصير واحد معك infection، مثلا واحد صار معه upper respiratory tract infection، you have cells in their exponential phase causing the infection. So you want to kill these cells in their exponential phase. This is why we want to study them in exponential phase. This is sometimes because this is where they are at their most virulent. هلا كل هذا حكيناه بنحكي عن خلايا بتعيش in a manner where they're constantly moving. and هلا إذا تترك الخلايا not moving, you do not change the moving conditions. What happens? There is a different growth mechanism for different, sometimes specific different bacteria. هدول اسمهم biofilm forming bacteria. So when they are stationary ما بموتوا أو بصيروا ما بيقدروا يتحركوا. Instead they start laying foundation حوالين نفسهم matrix زي ما حكينا بالمحاضرة الأولى of these glycoproteins or whatever. and then they start بخبوا حالهم جوا. هلا إذا بخبوا حالهم جوا هم راح يقللوا دخول النوترينتس على حالهم ممكن. فممكن they might lay dormant in this biofilm. هلا هم زي كانوا مغطين حالهم بشيلد so they cannot be destroyed with antibiotics with your immune system, but still they are still growing within the biofilm. ممكن كمان تحتيهم مثلا إذا كان تحتيهم gingiva أو تحتيهم tooth أو تحتيهم heart valve أو تحتيهم bone, whatever they might be, they might be having access to nutrients ومن فوق مسكرين على حالهم you cannot get to them. So this is a very nasty way for bacteria to maintain their communities. So they maintain بيضلوا يحكوا مع حالهم عنهم بعاد عن بعض جوا البايوفيلم، بيضلوا يحكوا مع حالهم and they can communicate and then keep causing disease. So that's why it's very important to bright our teeth every day. أهم إشي إذا بتشوف مثلا بالماوث maintaining water حركة مية اللي هو saliva prevents these biofilm from forming. فاللي بصير معه xerostomia مثلا بصير معه dry mouth دائما it will have gingival disease, gum disease. Why? Because bacteria start forming, they're not being sloughed off, they start causing disease. Brushing teeth is what is basically preventing bacteria from being dormant enough to form biofilms. That's basically so you're preventing this growth pattern for them. So this is a very different growth pattern زي ما حكينا. By forming layers of bacteria growth at the matrix, they are less susceptible to immune cleaners زي ما حكينا, antibiotics, chemotherapy, and unable to penetrate these layers. So the definition and measurement of death. What is death for a microbial cell? هذه النقطة كثير مهمة. بنرجع لبداية المحاضرة لما حكينا إنه my approach of destroying bacteria is flawed. شو هو قتل البكتيريا؟ إذا بدي بسألكم مثلا هلا بدي أعمل لك اعمل pause بعد ما أسألك و think about it. What is the measurement of death? How do you measure تحكي إن البكتيريا هذه ماتت؟ So it is losing its ability to reproduce. هلا مش حكينا إذا كان عندي 10 مليار خلية وأعطيتهم كل الضغط بالعالم والأنتي ميكروبيالز والحرارة whatever, and one bacteria survived and it was still able to reproduce, بعد أسبوع ترجع لي كمان ترجع لي أقوى من أول. So if it still has the ability to reproduce, it's not dead. مع إنه قتلت الخلايا كلهم، قتلت 10 مليار خلية، بقول لك قد ما بدك رقم قتلتهم كلهم. But if you have not stop them from reproducing, you have not killed the bacteria. مع إنه, so it is not the destruction of cells, is the death, the measure of death of bacteria is their stopping their ability from reproduction. So a cell that is unable to reproduce on any type of medium is considered dead. معناته حطيتها أنت الخلية والخلية لساتها عايشة, has gene function, has proteins, it is eating, but is not able to reproduce. حطيتها على culture أو حطيتها على agar or whatever medium and it's not growing. مع إنها عايشة, it's not reproducing, we consider it dead. Because this is how, يعني هلا بلشنا نفهم البكتيريا أكتر. So however, this means that the choice of medium as well is important. مثلا أنت حطيت هاي الخلية العايشة بميديم بس ما بتقدر تستخدم الميديم هذا, ما بتقدر تستخدم الشجر اللي حطيتها ولا البيبتون ولا whatever. It cannot use it, so it did not reproduce. فأنت رحت, you went and reported that this bacteria is dead because it did not reproduce. It's not really dead, but you used the wrong medium. إذا حطيت الميديم الصحيح, it will grow. So choosing the correct medium, choosing the correct conditions for growth is يعني إذا حطيتها in the correct conditions and وما عاشت, هاي ميتة. بس إذا حطيتها in the wrong conditions وما عاشت, it's not necessarily dead. So this maybe represent the following. We played out there's no growth, but it could also mean that there's inhibitory substance in the sample itself, مثلا, which prevents. This is another example. So once diluted the sample, we reach a low enough concentration of the inhibitory substance in the sample that would allow microbial cells to grow. تمام. The incubation in the first hour of treatment is also critical. مثلا إذا أنا حطيت خلايا bacterial cells and you irradiate them with ultraviolet light وحطيتهم على طول, you will find that most of them are dead. بس إذا أعطيتهم دقيقة, أعطيتهم مثلا 20 دقيقة in a buffer ياخذوا نفسهم, you will see that only 10% died, مش 99% died. Why? Because you have given them a chance to reproduce again, you have reduced the stress. حطيتهم stress ماتوا كلهم. حطيتهم stress وعطيتهم فترة ياخذوا نفس وحطيتهم رجعتهم على الstress الثاني, you will find that 90% have survived instead. So conditions are important. Timing is also important. So a microbial cell that is not physically disrupted is thus dead only in the terms of conditions used to test its viability is very important here. So the measurement of death is similar to measurement of growth. Measuring death is done on the whole population of organisms and not on an individual cell. So the condition causes death of 90% to die. The probability of one cell dying in a 10-minute interval is 0.9 مثلا. So this is self-explanatory. هلا شوف الفرق بين هدول التنتين. This is a death curve and this is another logarithmic death curve. So they start dying here and then they start dying here. Why are we studying death curves? Because these curves basically هم الكيرفز اللي احنا بندرسهم لما نشوف إذا عندنا antimicrobial شغال ولا لا. We have a mechanism to destroy bacteria. We want to study it, bacteria, viruses, whatever. If you want to study them, نشوف بموت ولا ما بموت. This is what we study. So what happens here? شو الفرق بين هاي وهاي? Both of them start at 6 but هنا في عندي shoulder. في عندي lagging before they start dying. Here they start dying right away. What is the difference here? In the previous figure, the A pass of typical inactivation event when many antimicrobials are used periodically as observed. Should difference between A and B اللي هو الشولدر هذا. This is because the killing was immediate in the first at 0 time. ايش يعني؟ شو الفرق هلا هون؟ إذا في عندي شغلة البكتيريا عندها منها كوبي واحد. مثلا شو عندها بكتيريا عندها منه كوبي واحد؟ جينوم. It has only one genome. So if I inactivate this one thing and it does not have anything else to rely on, it will start dying right away and start going through the phase right away. If I target something that the bacteria has many copies of, مثلا ribosomes, if I started, so at the beginning I عندها مثلا مليون ribosome, I give her a drug that kills ribosomes. So أول شيء قتلت 500 لسه they're not dead. قتلت 750 they're not dead. قتلت 900 they're not dead. خربت لهم كل الريبوسومز. Now they start dying. That's basically, so this is many hits before death. This is one hit before death. So that's basically what it is. So this means that a single hit by inactivating agent is sufficient to kill the cell, and the vital target اللي هو مثلا DNA, membrane, chromosome هذولا is something that does not have multiple copies of مثل الانزيم or ribosome. So that's the difference between the two. So هذا بدخلنا على sterilization. What is sterilization? Is for this purpose we study these environment dynamics because we seek to achieve sterility. I think let me see how long if this gone. Okay, so let's stop here and then we'll continue محاضرة الجروث الثانية in the next lecture إن شاء الله.